Okay, Chapter 2, Part 2. This is the study of biochemistry. And when we're looking at biochemistry, this is important to remember that it's going to be slightly different than everything we've talked about. It'll go a little bit more in deeper. And what's important about biochemistry is this definition right here. It is the study of chemical composition and reactions of living matter. So what we're going to be focusing on is things that are going to be considered organic compounds in biochemistry, and organic compounds are going to contain carbon. So some sort of carbon molecule, and they're all covalently bonded. Whereas when we're talking about inorganic, which we'll talk a lot about, these compounds do not contain carbon. And so while they are actually going to be bonds, they are not going to always be covalently bonded. This is typically going to be water, salts, and acids and bases. So as we move further, so looking at section 2.6, the focus here is going to be on water. So this is an inorganic compound. Water is the most abundant, and it is accounts for about 60 to 80% of our living things, for our volume of our living cells. Apologize. It is the most important inorganic compound because of these specific properties, and we're just going to run through the highlights of these, but it is very, very important. Water is also typically going to be the main solvent in your body. So remember when we talked about solute and solvent, that's the language we're going to be talking about. So some interesting, very cool properties of water. So these properties of water are what make it super, super important. So the first property is going to be the high heat capacity. And this high heat capacity is essentially going to be the ability to absorb heat. So it can absorb heat, but it doesn't actually change the temperature. So it's a really cool ability of water. It also allows for there to be changes in the external environment, but it will prevent changes in that internal environment. Another property of water is the high heat of vaporization. So this means is in order for water to turn into vapor, it has to hit a very, very high temperature. This is important because our bodies can get up to, during a fever, 103, 104 degrees Fahrenheit. You don't want your water to actually boil and turn into vapor. So there's going to need to be a large amount of heat. So that's important for our bodies to have. Third feature is going to be polar solvent properties. So this makes water the primary solvent in our water, in our bodies. So remember, solvent is the thing that is dissolving whatever is being put inside, and then the solute is being dissolved. So this is going to be the water, this is going to be the salt. You put them together and you are going to get salt water, but it is the water that is going to be the solvent. And it is important because it will actually be able to dissolve large ionic compounds. And this is a very important concept right here. It is the body's major transport medium. So things will dissolve in our water in our body and then be able to be transported around our body. So here's an example of that NaCl that we talked about last time. Okay, the next property of water is going to be reactivity. And this essentially means that H2O is a very important reactant. So it has the ability to react in chemical equations and be a really, really important part. Some reactions will actually require H2O in order for them to occur. And since most of our body is run with chemical reactions, water is very important. And then another feature of our property of water is cushioning. Water is actually a great place that will actually protect some of our organs. So there's actually going to be liquid all the way around your brain and spinal cord that the water will create a cushion in order to protect it. So that's very, very, very cool. Okay, so another inorganic compound. It's going to be salts. So when we're looking at salts, it is an ionic compound. So these are not shared electrons. These are just negative and positive that like to hang out with each other. So these are all going to be ionic compounds. And they're going to be specifically compounds that are containing anions other than a hydrogen ion. So a hydrogen ion has this little guy that he can donate, so he can actually give away that electron right there. So a salt is going to be something that's not a hydrogen, and it's going to have, cations other than hydrogen, and anions other than OH. So basically what this means is it is going to be not H2O. So when we're talking about a salt, it does not have hydrogen, nor is it going to have the OH. So it is going to be essentially something that is not H2O. So these are typically going to be called electrolytes because they can conduct electrical solution in the body. Some examples of these are going to be sodium, potassium, chloride. You can also have, I'm trying to think of magnesium. A lot of these are things that your body will actually eat. Oh, and iron, that's another good one. So things that you'll get from your food that are going to act as just the ions. So if you think about after a really heavy run, when the promotional items and the marketing that goes behind Gatorade and Powerade is being able to provide you with salts, you need something besides just the water. You already need the electrolytes in order to replenish your body. So that's what those are. Okay, this is really important for homeostasis, especially when it comes to maintaining their balance, which is the role of the kidneys. And if there is a disruption in any of this electrolyte imbalance, essentially all organ systems will cease to function. So as we get into later semesters and we talk about how things work, you will see chloride, potassium, calcium, all of these will show up again. And if they are not there, the system cannot work. Okay, we're going to do an overview of acids and bases. So if you've taken a chemistry class before, I hope this isn't new, but we're going to do an overview of acids and bases. These are both considered electrolytes, but they're going to be slightly different. So an acid will be something that releases the hydrogen ion. If it releases the hydrogen, this has a little positive sign. So it is going to be a proton donor. It's going to give. So in water, acids are going to release this hydrogen ion. So how much hydrogen ions there are is going to determine the acidity. So the concentration of the hydrogen ions, how much there is. So when you see concentration, it is done with these brackets that go all the way around. So that means quantity. How much of the hydrogen ion in the water will actually determine the acidity of something. So we're going to talk through some of the main liquids in the body and what their acidity ratio is. So concentration of the hydrogen ions is going to determine the actual acidity. That is different than a base. So a base is not going to release the hydrogen, it will accept the hydrogen. So that means that this is going to be a proton acceptor. And when this is happening, it is going to be the opposite concentration of hydrogen. So if you think about H2O, when this breaks up, you have one H and then you have OH minus because there's two hydrogen, and one oxygen. So this right here is the thing that can act as the acid or the proton donor. So whatever takes this will become an acid. Whatever takes this will become a base, because this is the proton acceptor. So as we're going through this, we'll talk about the levels of concentration and how these things work together, which is the pH. So this is going to be the concentration of acid and bases. It's very easy because they are just an inverse relationship of each other. So more hydrogen ions, more acidic. So it's going to be a floating scale. So the more hydrogen ions, the more acidic it is, whereas the more hydroxyl ions, so that's what these are, hydroxyl ions, the more basic it's going to be. So more acidic is related to hydrogen ions, basic is related to hydroxyl ions. And so this is a really, really, really important thing. So the more hydrogen ions in a solution, the more acidic that solution is. So remember that. So when we're talking about this, we're going to talk about something called a pH unit, and that will range from 0 to 14. So things that are going to be called acidic on that 0 to 14 scale have a high concentration of those hydrogen ions. So we're going to have a high pH. And then we're essentially going to have a low, sorry, not a high pH, low pH, high concentration of hydrogen, low concentration of hydrogen. So that's where we're starting. So if we have a high concentration of hydrogen ions, it will be a low pH. So that means on a 0 to 14 scale or a 1 to 14 scale, this is going to be a 1 if this is our scale. So if we have a high concentration of hydrogen ions, we're going to have a low pH. Same thing is going to be true for these guys. Basic solutions are going to have a low concentration of hydrogen. So that's going to be up here. Low concentrations of ion, and it will be a high pH. So this will be an acid, and this will be a base. So the scale goes from 1 to 14. So right in the middle between 1 and 14 is going to be 7.0. So what this is saying is that something that is basic is going to have a pH of 7.01 to 14. So anything that is basic is going to be on this side of the scale. It's going to be a basic. Whereas anything that is going to be not basic but considered acidic will be on this side of the scale. And it will be 1 to 14, or 1 to 17. And this will be an acid. So that's how the scale actually works. Acid is going to be 1 to 7.0, and this is going, or 1 to 6.99, right below it. 7 is considered neutral, and then this is going to be 7.01 to 14. That is how that process works. So you can see on this image right here, on the next slide, how that actually plays out. So when you're looking at this, remember that we said that 7.0 is acidic, is neutral. So that's going to be this right here. Neutral is 7.0. Right here is our blood, our main liquid in our body, besides that is made up primarily of water. So blood is actually going to be 7.4. So that means looking at this, that our actual body is going to be something that is going to be slightly basic, because 7.4 will live in this world right here, which is going to fall into our basic world. So our blood, our body, is going to be slightly basic. You can also call this as slightly alkaline. So if you've ever heard that term about alkaline water or the pH of your body, that is what it's referring to. But you can see some of the other things in our bodies and where or household items or other pieces and what those pHs are. So what we care about with a pH is something called neutralization and buffers. So we're not going to actually dive into any of these this semester. This is going to primarily come into play second semester with some of those physiology pieces and those systems. But with a neutralization reaction, if you actually mix these together, if you mix an base and an acid together, you are going to get water and something that is essentially neutral, a salt. So you can actually add an acid and a base and it cancels each other out. So that's a neutralization. And then a buffer, what a buffer will actually do is it will keep the pH, if we've got a scale of 1 to 14, a buffer will actually keep it from going from way down here to way up here. It will stop that process, and it does that by keeping things, by releasing and adding and breaking and removing hydrogen ions. So it just kind of rotates through that. Again, this will be covered in AMP2, but you just need to know the term buffer relates to acid and bases. Okay, the next section is 2.7. Looking at our organic compounds of synthesis and hydrolysis. So we are now moving into organic compounds. So all of the things we're going to talk about next are going to have C. They will all contain carbon. Everything that we're going to talk about now. Okay, so carbon is really important because it is electroneutral. It does not really gain or ever lose electrons, so it is just super, super, super stable. Carbon is unique to only living systems, which is why this is going to be biochem. It's the study of living organisms. There are 4 types of organic compounds, and we're going to talk about them each individually: carbohydrates, lipids, proteins, and nucleic acids. So we're going to start with, they all have the same foundation. So they all are going to have something called a polymer. And a polymer is going to be a chain of molecules. So thinking about a train car, you start with the engine. If you add more, you will get a chain of cars. So all of these are going to start with one type, and then you essentially can add more of that type. If you add it all together, you are going to get a polymer, which is the chain of molecules. But they're all going to start with something that's going to be a building block. This building block is called a monomer. So a polymer is a chain of molecules, but you can more correctly say a chain of monomers. So this is going to be one train car. You add them a bunch together, you're going to have a chain of the same thing. So that is a term. Polymer and monomer are terms you need to know. They are going to be put together by dehydration synthesis. So this is going to use water. And then you're going to break these guys down by hydrolysis reaction, which will give water. So basically that means down here is if we take monomer 1 and monomer 2 and we put them together so it's monomer 1 and 2 connected, you're going to lose a water molecule. Whereas if you have monomers that are already attached and you break them, so here's 1, here's 2, you've done it because you've used up a water, hydrolysis. Okay, we're going to give that water back to it. So here we go. Here's some examples. So this is super, super simple. These guys right here at the green and pink box. But you can see here how complex these pieces are. But all of these are going to really just relate to the interaction between a hydroxyl and a hydrogen ion in order to make a water. And then they will connect this way. So if you're unfamiliar with biochemistry or chemistry in general, at this level, whenever you see this shape right here, or you see this shape right here, this is a 3-dimensional ring is what it's called. Every single one of these little connection points is considered a carbon. So this would be carbon, 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 carbon. So this guy right here, carbon likes to have 4 connection points. So if you were really drawing it out, it would have 4 connection points. So carbon has 1, 2, 3, 4 points. So this would be a hydrogen, this carbon is attached to this carbon, and this carbon is attached to this carbon. So it has those 1, 2, 3, 4 connection points. But what you'll see in your book is it'll just be drawn like this but you need to know that every single one of these is a carbon, and it will have 4 connections. If there's nothing listed, it will automatically be a hydrogen. So that's how to read those guys. So coming in at 2.8, looking at carbohydrates. So we're going to go through all 4 of those types. So when we're looking at carbohydrates, it is going to be containing a carbon, a hydrogen, and an oxygen, and these will be in a ring. So these shapes will be in a ring, and they are going to have 3 different classifications right here. And these guys, these classifications, they are going to be classified by size. So small, medium, and large, essentially. So classified by size. Monosaccharide. Mono means 1, so this will be 1 sugar. Carbohydrate and sugar is synonymous. Disaccharide is going to be 2 sugars. So that's slightly bigger, whereas polysaccharide is going to be more than 2 sugars. So it could be 15, it could be 3, it could be 500. It's just 2 plus sugars. So 1 makes up, put them together, put that 1 monomer together, you get 2, that's a disaccharide. You put a bunch of those together, you get a polysaccharide. So we're going to break these down and start with monosaccharides first. So monosaccharides, what these guys are they are also called simple sugars. So they are made up of only 1 type of monomer. Woo. They are only made up of 1 type of monomer of a carbohydrate. These typically contain 3 to 7 carbons. But remember, these are going to be in a ring. So there are 2 really important monosaccharides. There's ones that are going to be called pentose and hexose sugars. So pentose means 5. So that is going to be 5 carbons. A hexose means 6, hexagon, so it'll be 6 carbons. So the way they're organized will be different, but they are going to have 5 carbons or 6 carbons. So coming down here to our list of monosaccharides. If you look here, we've got hexose sugars. Here we've got pentose sugars. So these are going to have 6 carbons, and these are going to have 5 carbons. So remember, you're counting the little dots, 1, 2, 3, 4, but you're also counting if there's any actual carbon. So monosaccharides. There are 5 different monosaccharides that you need to know. These are very, very important. And these monosaccharides are the foundation for all sugars and all starches. So there is going to be glucose, which is this guy right here, fructose, which is this guy right here, galactose, and then you're going to have deoxyribose and ribose. So these are the 4 building blocks of a sugar: glucose, fructose, galactose, deoxyribose, and ribose. You do need to know these by sight. These are super, super important, and you will need to know these for the rest of it. A&P. If you put these 5 important monosaccharides together, I can put a glucose with all of these. I can put glucose with a bunch of itself. You can add them all up because remember, they're building blocks. So when you do a disaccharide, there are some very common combinations of these 2. These are going to be double sugars, so it's 2 monomers. These are decently big, so they're too large to pass through cell membranes. And the important ones you need to know are sucrose, maltose, and lactose. So when you're looking at these, the disaccharides, and I believe this is actually, depending on the edition of the book you are using, has been wrong in several editions on which disaccharides are named which. So just to double check, the edition has been updated. Okay, so here are our disaccharides. Sucrose, I said, is one of them. When you look at sucrose, this is going to be made up of glucose plus fructose. That is how you get sucrose. Glucose plus fructose equals sucrose. When you have maltose, this is going to be made up of glucose plus glucose. So it's going to be different. It's glucose plus itself. When you do glucose plus glucose, you get maltose. And then the last one is going to be lactase, or lactose, I apologize. And lactose is made up of glucose, but it's also made up of galactose. So with lactose, glucose is going to be the foundation for all of them, but this one you are only adding galactose to it. So that is how these two go together. You need to know these as well. These are very, very, very important. Okay, so looking at a polysaccharide, when we add these guys all together, and we have a lot of them, these are going to be very big, very, very, very important. And there are two important ones, starch and glycogen. Starch is actually going to be this carbohydrate storage that you're using and you eat, but it's specifically going to be plant-based. So starch is plant-based polymer or polysaccharide, whereas glycogen is going to be the animal-based storage of polysaccharides. These are very big. They're not very soluble in water, but they are very delicious to eat. So here's some examples of some polysaccharide. This is glycogen, which is the storage in animals. And you can see how each one of these is a monomer. You put them all together and they're going to create a polymer, but you can also see how they're all rings, because remember that is a signifying fact of a carbohydrate. Okay, moving down to the next one is going to be lipids. For lipids, these are also going to contain carbon, hydrogen, and oxygen, but they're going to be less than in carbohydrates. It's not going to be as much. And there are a couple main types. We're going to focus on the first three and not spend a lot of time on eicosanoids. So looking at these lipids, they are also going to contain carbon, hydrogen, and oxygen. So lipids are considered neutral fats, but when you see a lipid, also lipid is synonymous with the word fat. So we're going to use that word quite a few times when we're talking about lipids. So these do not dissolve in water, and we're going to start with triglycerides, the first type. So triglycerides are neutral. When they are going to be solid at room temperature, they're considered a fat. When they are going to be liquid at room temperature, they are considered an oil. So this is why your coconut oil can be either in the solid form, which coconut fat, or it can be in the liquid form of coconut oil. So these guys can change based on the temperature of the room they are in. These are very, very large. They are composed of hundreds and hundreds and hundreds of atoms, but they are very, very important because they are the best form of stored energy. So this potential energy that our body has, this is the best form of stored potential energy. And there are the triglycerides are made up of two parts. So they're going to be made up of something called a fatty acid, or a saturated fatty acid, as your slide says. And a fatty acid is going to be linear chains. So it's going to be chains that are in a line, and they are going to contain carbon, hydrogen, and then end with a carbon, oxygen, oxygen, hydrogen. So it's going to be a very fat chain. You'll see it like this. And then it'll end with OOH, or COOH. And then it will also be made up of a glycerol. So a glycerol is a simple sugar. So when we put these two things together, a fatty acid and a glycerol, I'm going to come down here to this guy. Here's our glycerol and our fatty acid. So we take our, essentially, it's called a backbone, our glycerol backbone. So if you take this glycerol backbone, and remember, this is called a triglyceride. So these are tri or 3 fatty acid chains. So here's our here's little carbon, 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 and coming off of it, here is my fatty acid chain. Here is my fatty acid chain. Here is my fatty acid chain. So this is what it looks like, where it'll be made up of a glycerol backbone and then your 3 fatty acid chains. That's how you get the triglyceride name. And those are the best form of storage. So remember that guy. Okay, another type is going to be called a phospholipid. A phospholipid is also considered a modified triglyceride. And how it's modified, it is only made up of 2 fatty acid chains. So it will still have the glycerol backbone, but it will only have 1, 2 fatty acid chain. So it's a modified triglyceride. With this, it's really, really cool because this right here is going to be considered the head, and then all of these pieces are going to be considered the tail. And they have different properties. The head is going to like water. We'll talk a lot about this in chapter 3. Whereas the tail is going to hate water. And those 2 properties alone are really important when we get to chapter 3. So here is your, and they also, I apologize for not adding this, they are also going to contain a phosphate group. That's where the phospholipid will come in. So here we're going to have our phosphorus group, your glycerol backbone, and then the 2 fatty acid chains. So that's really important. The next type is going to be a steroid. And this is always going to be 4 interlocking rings. So a steroid will be made up of 4 interlocking rings. It is considered a fat, a lipid. It is not a carbohydrate. So some common steroids, you can see right here, we've all talked about these: cholesterol, vitamin D, steroid, and bile. And these are really important. So the most important steroid is going to be this cholesterol one, because it is responsible for vitamin D, plasma cell membrane, a lot of things we're going to talk about this semester. But just to give you an overview of what it looks like, here is 1, 2, 3, 4 interlocking rings. So when you see that, you will automatically know that that is a steroid. And eicosanoids, we're not going to spend a lot of time on them, but I just want you to know the name. It does fall into a lipid category. So there's a bunch of different kinds. It is a derivative of a fatty acid. It is also found in cell membranes. And when you get to blood pressure and all that, it'll come up again, but then also in inflammation and labor contractions. So we're not going to look at that one, but you do need to know where that one lives. Okay, so we've done carbohydrates and we've done lipids. Now we're going to do proteins. Proteins are really important because it is going to be 20 to 30% of our overall cell mass are going to be made up of proteins. These are going to contain, like everything else we talked about, carbon, hydrogen, and oxygen, but it will also contain nitrogen, and sometimes sulfur, and sometimes phosphorus. But these are the primary ones. Nitrogen is new. These are also going to be made up of monomer, that if you put a bunch of monomers together, you are going to get the chain that is the polymer. It's going to follow that same process, but these are going to have different names. So this monomer right here is going to be called a peptide. So the peptide is the monomer of the protein. And you put them all together, and it's not just going to be one long line like this. There will be different types of structures when they are all put together. And there are 4 different levels of structures when they're all put together. but the monomer is the foundation. All proteins are made up of 20 different types of amino acids. What does that mean? So, amino acids are going to be an acid or a base, and they're going to be connected by peptide bonds, but this guy right here is going to be an amino acid. So essentially, it is going to be an amine group, which has a nitrogen on one side. It's going to have a carbon in the middle, and it's going to have an acid group, which is usually going to be COOH if you shorthand it. So you're going to have an amine, you're going to have an acid, and then this carbon right here will be attached to something called a functional group, or an R. And that functional group right here is going to be, there's 20 different types based on whatever that functional group is. So looking down here, here's our amino acid with functional group R and functional group R. You put this together, it's not an amino acid or a monomer and a monomer. It will now be called a dipeptide when you put them together. But this R right here and this R right here are going to be different. So these are really cool. These R's are these functional groups. You're going to start thinking about them as letters. So if this one was the letter A and this one was the letter T, you put it together and you're going to actually spell the word at. So these amine acid functional group combinations are what gives proteins their structural importance in the body. And so, like I said, there are going to be 4 different structural levels that when we put these guys together, you're going to have the monomer of a protein, which is the amino acid. You're going to put them a bunch together. You're going to end up with a protein. So here we go. The structural level of a protein. There are 4 different types. And each type will determine what the function of the protein is. So when you have a primary structure, or a 1 primary, 1 degree, this is going to be a linear. So this is going to be a chain of line 1, 2, 3, 4, 5. They are just in a line, straight. The second type is going to be a secondary. And this is going to be a folded line. So with this secondary, it'll either be folded as something that is called a alpha helix, which is going to be a coil. You can fold this line, think about a string, you can fold it so it does this. It's coiled. Or you can fold it into something that's called a beta pleat or a beta sheet. That will be essentially, it's pleated. So here's one, and it goes like this. It is folded in this direction. So you can see how those two are essentially, if you were to take a string, you can either coil up that string or you can pleat it and fold it this way. But either way, the secondary is a folded. And when these are happening, you actually have interactions within the protein itself based on how it is folded. The third structural level is tertiary, or 3. And this is going to be how the alpha helix and the beta pleat fold on itself. So this is going to be a blob, essentially. It is going to create a globular protein. And this globular protein is going to be both alpha helix plus beta sheet. It is a blob. So before I go on further, I want to go through these pictures because they show you what's going on. Here's the primary structure. It is 1, 2, 3, 4, 5 in a line. Here's the secondary structure. Alpha helix, twisted, beta pleat, where it is actually pleated together. And then if you put them together, you get something called the tertiary structure. And here you can see there's a coil right here, and then this right here is a pleat. So it's got both, but it essentially creates this big blob. It's a globular protein. So then the fourth kind, I'll go back up here to keep them together, is called quaternary, or the fourth degree structure. This is going to be 2+ polypeptide chains, 2 or more. When you put essentially a quaternary, a tertiary with a tertiary, you're going to get a quaternary. So here's one blob, here's a second blob. They are both at the tertiary structural level, but you put them together and then they are combined into the quaternary, where it's 2 or more polypeptide globular proteins put together. So it's very important. Now remember, at this point, the structure of a protein determines its function. So proteins will either fall into one of 2 categories. They can either be something that is called a fibrous fibrous protein, and this fibrous protein is going to dissolve in water, which is important, and it is very, very, very stable. And it's essentially going to provide structure in our body. So it's responsible for keratin. It's not responsible. It is called keratin. It can also be a form called elastin or collagen, or some contractile fibers in the body. But for women who are listening to any kind of beauty ad out there, if you've ever come across one of those, they talk about elastin, collagen, and keratin as a structural protein in the body. They're talking about essentially a fibrous protein. The second type is a globular protein. These are also going to be a functional protein. So because it's globs, they are very, very compact. And these are going to have specific functional areas or functional regions called sites. Some examples of these are antibodies, hormones, some enzymes, and some other things. So if this is my globular protein, what that means with specific functional regions is this guy right here is the only place that's going to interact with something else. So this right here would be its active site. That is very important. As we get later in the semester, we'll talk about how proteins relate only with other proteins or other things at their active site. So here's one of the biggest concepts of this chapter is protein denaturation. Protein denaturation. Essentially what this is if you can unfold or lose the shape of a 3D protein. So if you take everything that we just talked about with a protein, a globular protein that's all folded and twisted and crumbled together, if you lose the shape of it, you will actually lose the overall function, because remember, structure determines function. So in this particular case, you lose the structure, you lose the function, because you lose that active site right here. It's no longer able to be utilized. How you can denature a protein so much so that you unfold it and it loses its shape, you can do this by increasing the temperature, or you can do this by decreasing the pH, which is also increasing acidity. So if you think about any time you've boiled something, let's say boiled eggs, if you increase the temperature, you're going to denature and change the structure inside that egg. And you've done that because you've boiled it. Or the opposite is if something goes into hydrochloric acid, it will dissolve because it's losing and unfolding that actual integrity of the shape. Both of these are not reversible. Once this happens, you are not allowed to go back and fix it. You have completely destroyed the shape of the protein. If you destroy the shape of the protein, you lose its function. This is what happens in our body, where you lose the function of something. We see it. It's a physiological issue. There's sickness. There's something going on. But the reason that happens is because the actual structure of the protein has been compromised. So that's an important concept. Another concept is going to be enzymes. These are proteins. They're specifically globular proteins. So an enzyme is a globular protein, and it will act as a biological catalyst. So a catalyst or a biological catalyst means that it is something that will regulate and increase the speed of the chemical reaction, but it will not be used up in the process. So you add the catalyst to a reaction, but you also get it on the other side. So this is important because it will lower the energy needed. So here's a couple drawings that really show this. So here is without an enzyme. It's the first graph. You redraw that line. Without an enzyme, your energy is going to be high up here and low down here. And on the x-axis, it'll just be time, start to finish. It just happens. So this is going to be your reactants. It's the beginning of your reaction. Remember, think about the chemical equation. So a reaction requires energy to be used in order to happen. So you're here, the reaction hasn't started, the reaction starts, it requires energy, so the energy goes up. And then after you finish the reaction, the energy level will then drop because the product, the finished thing, does not need that high level of energy. So the difference right here is whatever this is the energy used to create the reaction. So the height, the amount of energy used to create that reaction is what is essentially the foundation of every time that reaction happens, you need to have that much energy. But what a catalyst, a biological catalyst, the enzyme will do is it will decrease that energy. So with an enzyme, I'm going to start at the same place because it does not change the chemical reaction, and I'm going to end at the same place. Still going to be low energy and high. But as I start my chemical reaction, I'm going to use some energy because you have to, but I don't have to use up much. And then I'll end up here. So what happens when you add an enzyme is you essentially decrease how much energy is needed for that reaction to occur. And that is going to be called activation energy. So the slide before, we'll talk about activation energy is the energy that is needed to initiate and complete a chemical reaction. So with an enzyme, it is a lower activation energy. So that is what is happening with that. So here you can see, I'm just going to skip to the last one. They're all the same. Here's my enzyme, my active site. These guys come in. They're going to create some sort of chemical reaction, chemical equation. The product pops out. When this is happening right here, this is the energy transfer and the reaction and the activation. Everything is happening at this step. But you'll notice that enzyme was here first. It has the chemical reaction happening. It's 2 amino acids. Here it's a dipeptide, but the enzyme is left at the very end. It's still there. It's not used up. But this is that activation energy. It causes that to be less. Okay, we have two more topics, 2.11 and 2.12 for this chapter. 2.11 is going to be covering nucleic acids, specifically DNA and RNA. So these are all going to contain carbon, hydrogen, oxygen, nitrogen, and phosphate. So as we talked about earlier, with some of them only containing carbon or hydrogen, remember we talked about carbohydrates, lipids, proteins, and now these are our nucleic acids. So the monomer of a nucleic acid is called a nucleotide. So you're going to put a bunch of nucleotides together and you're going to create these nucleic acid. So this nucleotide is there's going to be a couple different kinds. There are two classes of nucleic acids, and they are going to be DNA, which stands for deoxyribonucleic acid, and RNA, which stands for ribonucleic acid. So DNA and RNA. You do need to be able to spell and know deoxyribonucleic acid and ribonucleic acid. So we're going to start with these. DNA. What is happening in this? This is our genetic blueprint. This is important for all of our who and what and makes us up. It is going to be double-stranded, have specific nucleotides. So what I'm going to do is I'm going to take these two charts right here and I'm going to put a combination between what is a comparison between DNA and RNA. We're going to talk about the comparison between them both. So when we're talking about these comparisons between the DNA and the RNA, the first thing we're going to talk about is the nucleotide. What is the foundational piece that makes up the DNA versus the RNA? So when we're looking at the nucleotide, we're going to compare DNA and RNA to these, what types of nucleotides they are made up of. DNA nucleotides are going to be these guys right here. You're going to have adenine, guanine, cytosine, and thymine. So the nucleotides are going to be A, G, C, and T. That is the shorthand. Nucleotides for DNA, adenine, cytosine, and thymine. We're going to come down here to RNA. And RNA is going to have, where is it listed? It's actually not listed on here. That's very interesting. Okay, so RNA will actually have different nucleotides. It will also have adenine, which is A. It will also have cytosine, which is C. It will also have guanine, which is G. But it will not have thymine. It will have something called uracil. So this is the difference, is that RNA will have uracil as a nucleotide, where DNA will have thymine as a nucleotide. But they both come with their pairs. So these are going to be complementary nucleotide pairs. They are both going to be able to do that. So with these, it is going to be adenine will always pair with thymine. Guanine will always pair with cytosine. Over here in RNA, guanine also always pairs with cytosine, but adenine will pair with uracil. So these come together. So that's going to be the first between the two. DNA and RNA have different foundational nucleotides. Another difference between the two is the function. Why we actually care about these guys. The function of DNA is going to be genetic material. So it's giving us the blueprint, and it's also going to be instructions for proteins, specifically protein synthesis. So the development of new proteins is going to be found in DNA. The function of RNA is going to be actually to do that protein synthesis. So it will actually build the protein. The RNA will build, but the DNA has the instructions to build. So that's the difference between RNA and DNA from a functional standpoint. They are also going to be different in their location. DNA will always be found in the nucleus of the cell, while RNA will always be found in the cytoplasm. They're going to be found in different places inside the cell. And then there's two other differences between them. The structure. So how these nucleotides are put together. The structure of DNA is going to be a double-strand helix. So here's my double strand. It looks like a ladder. And if I put it together, it'll look like a double-strand helix, just like this. Whereas my RNA will not be double-stranded. It will be single-strand. and it won't be a helix, it will be straight. So DNA will be like, RNA will be like that. So one is double-stranded and is twisted, one is single-stranded and is straight. And then they will also differ because they will have a different sugar that is going to be part of their formation. DNA, sugar, I'm going to go all the way up here, is going to be deoxyribose. See that as part of the name, deoxyribose. Whereas RNA will have ribose. So the difference based on their sugar is actually in the name. DNA will have deoxyribose as its sugar. RNA will just have the sugar of ribose. So looking at the two, so what I did is I just combined all of these into a couple different comparisons for charts. So here's your DNA. And you can see with this DNA, you have the sugar deoxyribose right here. Here's your base, adenine, thymine. And what you're also going to have is you're going to put the A with the T, which is what is always going to happen up here. Cool. So you're going to have sugar deoxyribose and a base and a phosphate on one side. And then on the other side, you're going to have the same thing, phosphate, sugar, and the base. And these two come together. And here is your twisted double helix. So that's DNA. When we come out and we look at our, I guess we don't have a picture of RNA. That's tricky. When we look at RNA, it will not quite look like this. We're going to talk more about this in chapter 3 when we're actually going to code some DNA and some RNA, and so that's going to be super exciting. So we'll see this again. This is an overview of those two. Okay, the last section of this is going to be ATP. The reason we need ATP is because right now we're going to flash back to the very first thing in the chapter, and we're going to talk about energy. So glucose, which we talk a lot about in the world of human physiology, is the most important cellular fuel. But it does not have enough energy. So as you use up glucose, it does not have enough energy to make reactions work in your body at the level they need to. So you can have glucose, your body will use it, it will break those bonds, it will, remember glucose is a polymer, glucose is the monomer of a starch or a sugar, and it's really, really important when we break those bonds, but it doesn't have enough energy. So the way our body will actually do this is it will use something called ATP, which stands for adenosine triphosphate. So adenosine triphosphate, ATP. You also need to know that one. This is going to be the primary energy transferring molecule in the body. So ATP is the primary energy transferring molecule in the body. So this is essentially what allows us to have energy and go. So ATP, this works because it will actually transfer a phosphate. It will, or transfer a molecule. And so when it does that, it will break a bond. And as it breaks a bond, because it's transferred a molecule, you are going to get energy released. And then you can actually use that energy. So it's a very cool process. So we're going to draw this ATP molecule right now. ATP has three things. It's going to have the adenine. It's going to have a ribose sugar. And it's going to have 3 phosphate groups. So triphosphate, adenosine triphosphate. So this is what you have right here with our picture right here. Here is our adenosine, adenine. Here's our ribose, and here's our 1, 2, 3 phosphate groups. And the way I know that these are 3 phosphate groups is because there is the phosphate in the middle, and that is what the phosphorus. So here's 1 phosphate 2-phosphate, 3-phosphate. And what we're talking about breaking is the space between them. So when you have 1, 2, 3 phosphates attached, it is going to be adenosine triphosphate. So you're going to have the 3 phosphate groups. But you can also have something called ADP, and that's going to be adenosine diphosphate. And with adenosine diphosphate, you have essentially taken these guys and recolor them in 1, 2, 3. And what you've done is you have broken this bond. If you break this bond, you're going to have a phosphate group pop off, and you're only going to have 1, 2. That will give you adenosine diphosphate, which is this guy. You can also lose one more. You can break another bond and have another phosphate group pop off. And if you do that, you're going to not be tri or di, you are going to be mono. You will be adenosine monophosphate, or AMP. Adenosine monophosphate. So you really only do ATP to ADP, usually is what's happening, but you have the ability to lose up to lose 2 phosphate groups. So the terminal group of the phosphate of the ATP, so it can be this guy right here as the terminal, it can be this as the terminal, can be broken, that bond can be broken, and then that will actually release energy to use your work. And then you simply just add another hydrogen bond. So then you can just add this guy, sweet guy, back up. So if this is going to be my phosphate, my nucleotide, I'm just going to draw it. He's connected to my ribose sugar. And coming off my ribose sugar, I'm going to have 1 phosphate, 2 phosphate, 3 phosphate. This right here will break, and you're going to get energy. This guy right here can break, and you're going to get energy. But if you have just, here's my nucleotide, here's my ribose. If I only have this, and I have a ADP, so it's diphosphate, I can actually add this phosphate back in. And so you can go from ADP plus the phosphate, and you can go back to ATP. So you can add and remove the terminal phosphate group in order to get that energy. So it's the terminal phosphate group that can be added or lost. So that is what's happening, and there's a lovely image here at the bottom. We're going to talk about transport in chapter 3. We're going to talk about mechanical with muscles in chapter 8 and 9, and then we're going to talk about the other one in some of the other chapters. So we will be hitting all of these as well. When you're reviewing and looking at chapter 2, we talked a lot about monomers and chemical or organic compounds. You do need to know by sight the 5 carbohydrate monomers. You need to know ATP and ADP. You need to know DNA and RNA. You need to know what a lipid looks like. You need to know what a triglyceride versus a phospholipid looks like. And you need to be able to know what a protein looks like. So, I mean, you don't need to draw them, but you do need to know what they are made up of and what their general structure looks like. And then ATP and denaturization of the protein and the levels are a big, important concept in this chapter. But that is the end of chapter 2.